We've had a good break. It's time for us to move on and focus on student presentations and our technical demonstration by Nikon. I'd like to invite Dr. Arthur Cheng, who's chairing the session, to share his screen and take us through this session. Arthur?
Great, thanks, Dave. Yeah. Okay. Everyone see my screen? There we go.
Yep.
All right. So I'm going to lead the next session. I'm chair of the next session of presentations. My name is Arthur Cheng from York University. And we have 4 student presentations, which are going to be 3 minutes each, followed by a technical demonstration from one of our sponsors, Nikon, which will be given by Dr. Sally Miller. So I'd like to introduce our first speaker, which is going to be Nicole Baker from the University of Ottawa. So Nicole.
Hi, everyone. So I need you.
You have to stop sharing and then let Nicole share, I guess.
All right, here, stop share. There you go.
Okay.
So hi, everyone. I'm Nicole, and I'm a master's student at the University of Ottawa. And I'm going to talk to you about how mitochondrial structure can regulate muscle stem cell fate and identity. Skeletal muscle possesses the remarkable capacity to regenerate, and this is largely due to the presence of a muscle stem cell pool. But often in degenerative diseases and in aging, this muscle stem cell pool gets depleted, though the underlying cause of this decline is not well understood. Mitochondria, which are our central metabolic organelles, have the ability to constantly undergo balanced cycles of fission and fusion, which is called mitochondrial dynamics. So often in degenerative diseases and in aging, we also see an imbalance in this dynamic process favoring mitochondrial fragmentation. But how mitochondrial fragmentation actually impacts muscle stem cell maintenance has not been investigated. So in muscle stem cells, we've observed that mitochondrial structure is highly dynamic. So using single myofiber cultures, we've observed that mitochondria undergo a transient state of fragmentation early in the activation process, followed by an elongation of the mitochondria as stem cells undergo fate decisions. So what happens if we disrupt this dynamic process in mitochondria and force mitochondria to remain fragmented, as is often seen in this disease context? Well, our lab uses a mouse model that conditionally knocks out OPA1, which is an essential mitochondrial fusion protein, and just the PAX7 positive adult muscle stem cells, which effectively causes fragmented mitochondria. So strikingly, in OPA1 knockout muscle stem cells, we get huge defects in many different aspects of muscle stem cell function and identity. So the first thing that we noticed was that OPA1 knockout muscle stem cells in a quiescent state have reduced levels of PAX7 mRNA. Furthermore, when they activate and enter the cell cycle, they do this much faster than their wild-type counterparts, indicating this primed state of OPA1 knockout muscle stem cells. And furthermore, OPA1 knockout muscle stem cells have defective fate decisions. So early in the regenerative process, OPA1 knockout muscle stem cells show reduced levels of PAX7 positive muscle stem cells with a subsequent increase in the number of MyoG positive cells, indicating premature commitment at the expense of self-renewal. And at the end of regeneration, so when regeneration is complete, the muscle stem cell pool should be fully replenished, but in our OPA1 knockout muscle stem cells, the muscle stem cell pool is not at all replenished. So this indicates a defect in the self-renewal capacity of these OPA1 knockout muscle stem cells, ultimately causing muscle stem cell pool depletion and failed muscle regeneration. So we can see that muscle regeneration in our OPA1 knockout muscle stem cells does not occur at all. So overall, our data provides us with a model that loss of OPA1 reduces the depth of muscle stem cell quiescence and enhances activation, while impairing fate decisions, ultimately leading to muscle stem cell pool depletion and impaired regeneration. And the data that we have uncovered here demonstrates a novel role for mitochondrial structure in the regulation of muscle stem cell identity, maintenance, and regenerative capacity. And it really paves the way for innovative therapeutic strategies to treat muscle wasting conditions. So with that, I'd just like to thank the members of my lab and my supervisor for all their help, and thank you for listening.
Great, thank you so much, Amber. Any questions, please post those to the question and answer box. See if we have any.
I have a question for Nicole.
Yeah.
Nicole, are these cells energy deficient? Like if you measure, if you knock out OPA1, do they still respire as well? I mean, some of the things that OPA1 might be due to energy deficiency.
Yeah, so this is a pretty acute knockout. So it's tamoxifen treatment 5 days and then knockout. And when we've done ATP assays to measure like ATP output, there is no defect. So they are still functional. So it's just really the fragmentation side of it.
Thank you.
All right, any more questions? All right, we're going to move on to the next speaker. If you still want to talk to Nicole, please answer your question into the chat box. Next speaker is Emily Hoffman from York University. Emily?
Hi, I'm just loading my screen. Sorry. Can you see my screen?
Yep.
But it's not full screen. Okay, is it okay if I just do it like this?
Please, go ahead.
Okay. So hi, my name is Emily Hoffman. I'm here at York University in the lab of Dr. Michael Riddell, and I will be presenting our abstract entitled Somatostatin Receptor Type 2 Antagonism Restores Counterregulatory Function and Confers Hypoglycemic Resistance in a Rodent Model of Advanced Type 2 Diabetes. Okay, so the prevalence of type 2 diabetes is escalating worldwide. And with that, there's a growing dependence on intensive glucose-lowering therapies to minimize the risk of long-term vascular complications. However, the therapeutic potential of these drugs is undermined by the risk of treatment-induced hypoglycemia, which is considered the major barrier to achieving optimal glycemic control. In fact, hypoglycemia is so common in type 2 diabetes that it costs the Canadian healthcare system over $65 million annually. Glucagon, as you can see here, is the pancreatic hormone that typically mediates the counterregulatory response to declining blood glucose levels. But in type 2 diabetes, this system is defective. We don't know the underlying mechanism, but it may be linked to another islet hormone called somatostatin, which inhibits glucagon secretion by binding to its type 2 receptor on the alpha cell. We hypothesize that the overactivation of this receptor suppresses the glucagon response to treatment-induced hypoglycemia in type 2 diabetes. So in this study, we sought to investigate the efficacy of the somatostatin type 2 receptor antagonist called PRL-2903 as a prophylactic measure against insulin-induced hypoglycemia in a rodent model of advanced or late-stage type 2 diabetes. So we induced type 2 diabetes in 18 male Sprague-Dawley rats through combined high-fat feeding and a low-dose intraperitoneal injection of a beta cell toxin called streptozotocin. So type 2 diabetic rats were subcutaneously injected with either the somatostatin antagonist or vehicle 1 hour prior to a bolus injection of fast-acting insulin to induce hypoglycemia, and we used healthy rats as non-diabetic controls. So here you can see our results. So starting on the left, somatostatin raised the blood glucose nadir out of the hypoglycemic range, as you can see here, from 3.4 to 4.2 millimoles per liter. And it also reduced the incidence of hypoglycemia by 40% in the treatment group. Here on the right, you can see that it delayed the hypoglycemic onset by 20 minutes, which was a significant difference. The glucagon response to insulin challenge, which diminished fourfold in the vehicle group, was completely restored with treatment, as you can see in our area under the curve analysis. And finally, on the right, immunohistochemical staining of islet tissue revealed a 70% increase in the expression of the somatostatin type 2 receptors on the alpha cells of type 2 diabetic versus healthy islets. So here we demonstrated for the first time that somatostatin antagonism reduces the frequency and severity of hypoglycemic exposure in a rodent model of advanced type 2 diabetes, which is essential for preserving cognitive function. And the relative abundance and potential activation of somatostatin type 2 receptors that we observed in these diabetic islets could offer novel insights into the mechanism of this somatostatin-mediated alpha cell dysfunction. So we propose that somatostatin antagonism as a potential adjunct to insulin therapy may be an important therapeutic tool for restoring optimal glycemic control in patients living with type 2 diabetes. Thank you.
Great. Thank you very much, Emily. Any questions from the audience?
Arthur, this is Tom Hawk here. I have a question.
Sure.
That's a very nice talk, Emily.
Thank you.
What causes the overexpression of SSTR2?
So we have no idea. This is the first time, to our knowledge, that this has ever been documented. We suspect that the islet remodeling following beta cell destruction, which we know to occur in type 2 diabetes, that usually involves the hypertrophy and or hyperplasia of the delta cells, which secrete somatostatin, and that could account for the increased expression.
So it's not necessarily an overexpression, but an increased expression of the delta cells? Is that?
It could, yes, it could be, but we will hopefully look into that in the future.
All right. I think that's all we have. Time we have. We need to move on to the next talk. Stay on time. Thank you very much, Emily.
Thank you.
Our next speaker is Amber Hutchinson from University of Guelph. Amber, please.
Hello, I'm just confirming you guys can see my screen okay?
Yep, looks good.
Perfect. Well, I'm Amber Hutchinson. I'm a PhD candidate at the University of Guelph, and today I'm going to be talking about my work that examines the dietary N3 versus N6 pufa and how they differentially modulate macrophage-myocyte inflammatory crosstalk. So starting with lean skeletal muscle, it's characterized by the presence of anti-inflammatory immune cells such as M2 macrophages, which promote normal metabolic function as well as insulin sensitivity. However, in obese skeletal muscle, is defined by chronic low-grade inflammation, which is defined by the accumulation of intramuscular triglycerides as well as immune cell infiltration and the presence of inflammatory cytokines such as TNF-alpha, IL-6, and IL-1 beta. Now, in obese skeletal muscle, we see a significant switch from M2 to inflammatory M1 macrophages, which are defined by INOS and CD86, as well as inflammatory cytokines. Now, these cytokines can promote the paracrine interactions of skeletal muscle, which can then cause metabolic dysfunction and can lead to insulin resistance and type 2 diabetes. Now, this inflammatory crosstalk provides us with a target for potential dietary intervention, specifically with the known anti-inflammatory long-chain N3 pufas. However, N6 pufas, are more prominent in our diet and are more controversial in terms of obesity. So our objective was to investigate the role of N3 and N6 pufa in the modulation of inflammatory macrophage-myocyte crosstalk in obese skeletal muscle. To do this, we had rats consume a diet of high-fat N6 or high-fat N3 for a period of 2, 8, or 12 weeks. We isolated CD11b macrophages from spleens and either co-cultured them with L6 myocytes and stimulated with LPS, or we cultured them alone with media derived from LPS-stimulated L6 myocytes. Now, looking at our results for macrophage-myocyte co-culture, we examined the gene expression of M1 polarization marker INOS, as well as the inflammatory cytokines TNF-alpha, IL-6, and IL-1 beta. With INOS, we see a significant increase with high-fat N6 compared to high-fat and high-fat N3 at 8 weeks. Whereas with our inflammatory cytokines, we see an increase in high-fat N6 expression compared to our other two diets at 12 weeks. Next, we looked at macrophage cultures with L6 conditioned media. And again, we examined our gene expression of M1 polarization markers, INOS and CD86, as well as the same inflammatory cytokines. Now, INOS and CD86 increased the expression with high-fat N6 compared to our other two diets at 8 weeks. TNF-alpha did the same with high-fat N6 compared to high-fat and high-fat N3 at 8 weeks. And then we saw a similar mechanism happening with IL-6 and IL-1 beta at 12 weeks. Overall, these data suggest that N6 PUFA could support macrophage-myocyte inflammatory crosstalk, in part by promoting M1 polarization. Further, these data provide mechanistic insight into the benefits of omega-3 instead of omega-6 to mitigate the development of inflammatory skeletal muscle dysfunction and ensuing metabolic comorbidities. Thank you.
Great. Thank you very much, Amber. Open for questions now.
Hi, this is Heather Edgell from York University. I can't seem to enter anything to the Q&A. I just have a quick question. In your data, you clearly showed that the PUFA 6, the N6, was bad for inflammation, but did you show any overt evidence that the omega-3s were good for inflammation? Or are you just saying that the omega-3s would be a good replacement for the 6s?
Here in this data, we are saying that N3s would be a good replacement in a high-fat diet as opposed to omega-6. So omega-6 is driving the inflammation that we see with a high-fat diet. So omega-3s would be better, but they do have anti-inflammatory effects, and the reason we're studying it is because we have seen those effects in other macrophage-myocyte crosstalk in other studies.
Thanks.
All right. Thank you very much, Amber.
Thank you.
And stop sharing your screen. Perfect. Now, and our next speaker is going to be Jaryd Te from University of Toronto. Please, Jaryd.
Great. Thank you. I'm just going to share my screen. Right. Can everyone see this?
Yes.
Okay, great. Good morning, everyone. My name is Jaryd Te, and I'm a second-year master's student working at the Keenan Research Center at St. Michael's Hospital. And today I'll be presenting my project titled Enabling Skeletal Muscle Repair and Functional Recovery Following Denervation-Induced Injury Using Ultrasound-Mediated Gene Delivery. The muscles in our hands and fingers give us incredible dexterity and enable us to perform complex and fine movements. From a surgeon performing a quadruple bypass, to a musician performing in front of thousands, to a father playing with his own children, our lives greatly depend on these muscles. However, long-term denervation injuries, especially those with delayed reinnervation, can result in the complete loss of muscles' regenerative potential and permanent functional disability. Why is this important? Well, denervation injuries occur throughout daily life, such as construction and motor vehicle accidents, gunshot or knife lacerations, and even newborn birth complications. Once productive members of society must now undergo significant rehabilitation as well as career restructuring. Some may even be unable to care for their loved ones, and some may even have to give up on their own dreams, say of becoming a surgeon or a musician. Unfortunately, there's currently a lack of practical or preventative therapies to treat this type of injury. So the aim of my project is to use the technique known as ultrasound-mediated gene delivery, or UMGD for short, to sustain the regenerative potential of denervated muscle. UMGD works by introducing DNA plasmids to the systemic circulation and then applying high-powered ultrasound to the target area, which facilitates the uptake and expression of DNA into cells. We delivered 3 genes, specifically IGF-1, VEGF, and ANG1 to denervated rat gastrocnemius muscle to the right hind limb. The left hind limb serves as an unoperated healthy control, and we analyzed samples after 14 days. So as shown in panels A and B, as compared to animals given empty plasmid, delivery of these 3 genes resulted in a mild increase in gross muscle weight, as well as a greater proportion of larger fibers. Furthermore, delivery of these three genes showed also an increase in overall vascularization of muscle compared to our negative controls. In the short time I've had, I hope I've demonstrated the great potential for umgd to be a targeted, non-invasive, and effective treatment strategy to combat denervation injuries. Perhaps one day this treatment will be the reason why a surgeon is able to perform that quadruple bypass, a musician is able to perform your favorite song, or why a father is able to play with his children again. Thank you very much. I'm happy to take any questions.
Thanks a lot, Lucas. I'd like to open it up now for questions.
Well, this is Tom Hawk. Interesting work. Is there any indication that inflammation or a inflammatory response is associated with these in your data so far?
So if I just understand your question, if the inflammation is associated with the benefits or the sort of the detriment of denervation itself?
No, just the delivery of plasmids and using the ultrasound to bring these plasmids into the muscle. Does this result in an inflammatory response?
Yes, for sure. So, well, the ultrasound itself is shown to sort of stimulate an inflammatory response, in addition to the denervation. So, one actual interesting area is the development of fibrosis in a long-term model. But we know in an acute injury, the inflammation actually really leads to sort of a regenerative response or a pro-myogenic response. And that's due to a progenitor population called fibroadipogenic progenitors. And that's sort of another angle of my project that I didn't have time to include. But for sure, inflammation and the inflammatory response, especially of M1 and later on M2 macrophages, plays a big role for sure.
All right, Jaryd, you have two more questions to answer in the chat box. Maybe you can answer those afterwards.
Sure.
But thank you very much for your talk.